Field Research virtual seminar series. It's been quite an interesting and enjoyable series of presentations, and I'm sure today will be a wonderful presentation. I'm happy to have Dr. Postava-Davignon here today, and she's going to give a presentation that she has called Adventures of a Termite Ecologist, Learning to Make the Most Out of What Nature Gives You. Before I turn it over, I'll remind you that there will be a discussion and question answer period at the end. In the meantime, your mics are muted. If you think you may forget a question, take advantage of the chat bar on the bottom to lodge that question, and we'll get to it. With that, I'll relinquish control to our speaker.
Thank you, Adam. So yes, while I work on sharing my screen, the first thing I would like to do is to thank Adam for inviting me to participate in this seminar series. As soon as I found out about it, I just jumped at the chance because this is a topic that is very near and dear to my heart. So I am thrilled to be able to tell you guys my stories today. And also to thank you to Lainey and John and anyone else at Lincoln Memorial that helped put this together. And thank all of you for being here. So what I will be talking to you guys about today, just to give you a little bit of background about myself, I am a self-entitled termitologist. I kind of shortened that from termite ecologist. And I've had the great fortune of having many field adventures in my time, and it was kind of hard to narrow them down. So what I would like to talk to you guys about today is a lot of work that I did primarily in my graduate school years and for a few years after graduate school at my main field location in the Virgin Islands. That was just one of the most transformative experiences for me, and it's definitely where I learned the most about being a field ecologist. So the photos you guys may notice during this presentation, I spent a lot of time down there, and grad school was now quite a few years ago. So you will see some trends in both the quality of photos I was able to take, but also some differences in my age. I like to pretend that I still look this young, but that's maybe not the case so anymore. But yes, what was I going to say? So the first thing I want to mention today that I think I actually forgot to include in my presentation, so I wanted to bring it up. But one of the most important things I've learned about field experiences are whenever one is presented to you, you must jump on the chance. So I was able to begin my work in the Virgin Islands back in 2004, a little bit by accident. So I worked in a termite lab at Northeastern University in Boston. And my primary advisor, Dr. Rebecca Rosengaus, had plans to do lots of field work in Panama, but that relied on quite a bit of funding, as research often does. And since that funding didn't come through right away, she wanted to provide myself and a fellow student an opportunity to get practice doing field work at a different location just until we had the chance to go to Panama in the future. So thankfully, my advisor was excellent at networking, and she happened to know my collaborator, Dr. Claire Fuller, at the time. She is now my collaborator as well, but I hadn't met her yet. And she enabled us to have the opportunity to go to St. John for a summer. So this was back in 2004. And long story short, it turned out that Dr. Fuller and I got along very well. We had very similar ideas in research, and it was the beginning of a very, very beautiful both collaboration and friendship. So I thought I would start off today just talking about some of the advantages and reasons why people bother to do field research. So why do it? For one thing, there are some very, very stunning locations out there, and I like to joke that termites live in all the best places. It's true. So I have had the great fortune of working in some very, very beautiful locations and traveling quite a bit for my work. You have the chance to get up close and personal with your study subjects and really get a feel for how they live and what their true biology and nature is outside. That is where the ecology happens. So even though you can learn a lot from lab studies as well, out in the field is where you can learn ecology best. And let's be honest, it keeps you in really, really good shape as well. Or if you did not stay in good shape, you are going to learn the hard way to get back into shape. So some lessons I've learned from many years of fieldwork. For one thing, fieldwork involves some funky outfits and some very interesting tools. So just in these photos, there was one instance where the place where we were collecting termites was a very fragile, unique forest ecosystem, and it needed to be protected from outside invaders, essentially, potential infections. So they made us wear these really fun plastic booties over our shoes, which was a very good time. And then in other places, you need to be aware of potential critters that are maybe not the ones you're interested in getting on you, such as ticks, potentially snakes wanting to get you. So you spend a lot of time with your socks tucked over your pants, which is very attractive, all that. And sometimes you get very, very dirty. So hopefully my students will forgive me for showing their very dirty bums after an excellent day of fieldwork. You will also encounter some creepy crawlies. Now, some of those are intentional, such as my termites. This is one of my favorite termites, a soldier on my hand, but others are not so intentional. So in one photo here, we have a very large banana slug. That one actually is from California, but on the island of St. John in the US Virgin Islands, one of the biggest hazards was the Jack Spaniard wasp, and they really liked being very cryptic. So you almost didn't notice them until you were right on top of them. And they also really liked hanging out by the termite nests, which was not very convenient. So do be aware of potential hazards. But even with those, sometimes the creepy crawlies are fun. Sometimes your study subjects may be a little harder to access than you would like. So termites particularly are very often in some very dense forests, in very thick groups of trees, but sometimes they are also a little bit high up on the tree, at least in the case of arboreal species. Now, what is depicted in this photo, I would not recommend for safety reasons, but sometimes when you just really want that sample, you do what you need to do in order to get it. I just needed a little bit of a boost to get into the tree, and that plastic blue chair just happened to be handy. Of course, my advisor at the time was not pleased with me doing that, but we did get the data, so there you have it. She told me she was going to tell on me to my mother. And last but not least, field time is finite and also very unpredictable. So you learn to work around the conditions you have. So this last one is one that I will be talking about throughout this presentation. But just to give some examples, for one thing, most people have a very specific field season. And so you have to design and plan your experiments around that specific season. And if something happens or something goes wrong, then you might lose an entire season of data, which can be a bummer, but it does happen. So I have known many field ecologists that have done a lot of work in a field season, but perhaps something went wrong or the data didn't come out very well, and they would have to do repeats. So field ecologists tend to have a little bit longer time in school than some other folks, but it's fun while we're there. Sometimes weather can be a barrier. There is one saying that there is no bad weather, only bad preparation. However, in some cases, when you're using instrumentation that really does not like heavy rain, then there is such a thing as weather that's not going to work out so well. And I actually have no pictures of that because, funny story, my camera does not work so well in rain. So one example I do have a short video of, and this is a very old video. I would also like to add that my field work actually started pre-digital camera age, which is kind of funny to think about right now. So some of these videos are not the best quality. But sometimes when presented with challenges in the field, you have to get creative. So one challenge that we had with our arboreal termite nests, even though they kind of look like just a big mound of dirt, they are actually very, very hard. Actually, let me rephrase. The older ones are very, very hard because the longer they are growing and developing and the termites are adding material to it, they're almost as tough as wood itself after a certain point. And for a lot of the studies I'll talk about today, we used data loggers that were about the size of a large credit card, a little bit thicker, and we needed to get them inside the termite nest in order to collect our data. So we needed a way to get inside these termite nests. So Dr. Fuller invented a tool and had someone help her build it in order to make a credit card sized hole in a termite nest. So this is one of my students. We called it the javelin because it had a rectangular metal end that we would stick in the nest, and it had a little bit of a point on it. And then it was a slide hammer on the handle that we would essentially hammer into the nest. And it was not an easy feat, let me tell you. I did my share of javelining in my day. And you can also notice that Jacoby, who is working very hard to get this javelin in, the termites also don't nest where you want them to. They nest where they want to, which is not always the best angle for us. So that just gives you guys some ideas of some of the challenges and creativity that we come across when doing field work. All right, so for those of you that may be a little bit newer to termites, I did want to provide a little bit of background information because I am also a huge lover and promoter of termites. I'm a little bit weird. So just to give you guys some framework, the insect world is decently sized. If you go to an entomology meeting, there's a good chunk of folks there. But then if you narrow it down to those folks that study social insects, it gets a little bit smaller. If you examine the folks that study termites, it gets even smaller. And then there are the groups that study termites for the sake of learning more about them as opposed to trying to kill them. And that is an even smaller population from there. So my research largely deals with learning more about their ecology and how they interact with their environment as opposed to better ways to eliminate them. So what are termites? They are descendants of subsocial roaches, and they are one of the eusocial insects. So unlike ants, bees, and wasps, which are the other more familiar, perhaps, social insects, termites are in their own order. They evolved along different lines. So they're a little bit different from those guys, but something that all eusocial insects have in common are that they have cooperative brood care. And I have an adorable photo of termite alates. And then within the circle, it's a little blurry, but that is a group of eggs. So and termites are also cool because they have both a king and a queen. The king does live, and they live happily ever after for the entirety of their lives. So they're very romantic. And they also have sterile workers. Well, their sterility is flexible, but they have workers and soldiers that help them out, and workers help with the brood care. So in this photo is of a broken open trail, and you can sometimes see the slightly larger workers with rounded heads versus the soldiers that in this species have a very pear-shaped pointed head. There's an overlap in generations within a colony. and also they have a division of labor. So some reasons why termites are very often misunderstood. So most people, when I tell them I study termites, they're like, oh, hey, I have these insects at my house. How do I get rid of them? Or if they know that they're termites specifically, well, they still want to get rid of them. So most of the questions I field most of the time are unfortunately about trying to get rid of termites. But fun fact, only about 371 or 12.5% are actually considered pest species. And only 104 of those are considered serious threats. Now, unfortunately, many of those occur in the southeast of the US. So sorry, guys. Sorry about the invasive Formosan subterranean termites. They are kind of a pain. But that usually only happens when termites are introduced to an area that they did not naturally evolve in, okay? So in most areas, termites are just naturally occurring in the woods, doing their normal function as decomposers. And also in many areas of the world, they are actually important ecosystem engineers, meaning that they provide important ecosystem functions. So in some cases, it's just the decomposition. In some cases, their nests, whether they be those arboreal nests or mound nests or subterranean nests, they change the landscape where they are. And it actually influences a lot of other species around them. So despite the negative connotations around termites most of the time, they are actually very important to the ecosystem. So my interest in termites, at least with regards to some of the work that I'll be presenting today, had to do with something I noticed when I was learning about the different species that we had in our lab. So termites, there are lots of different examples of ways that they nest, and I actually became obsessed with their nests very early on. But along some evolutionary lines, there is a trend going from more primitive on the left to more recently derived on the right, from nests moving away from being either directly within or very close to the soil, up to mound nests, and then arboreal nests are a more recent derivation of nests. So there are lots of pros and cons to both nesting strategies. And also they went from being nests that existed in a single piece of wood and were simply galleries excavated out of those pieces of wood, moving up to nests that are constructed by the termites. And these very large mounds, this is a mound nest from Australia, and then an arboreal nest from the tropics. But if anyone were to hazard a guess what they are made of, I wonder if you would know that that is essentially big mounds of spit and poop, as I like to say. So the termites consume planty material, whether it be wood or litter debris or some other detritus, and then they poop it back out again. And then they use secretions to kind of mold it all together. So in a mound like this, there may be anywhere from hundreds of thousands to millions of termites. And I actually did take this picture of a mound, and at the time I didn't study termites yet, but already I was very curious and really wanted to scratch it open, but it was in a national park, so I have a feeling they would not have enjoyed that. So, anywho, I noticed this trend of moving away from the soil, and something else you need to know about termites is that because of their close association with one another, they have a lot of close relationships with microbes such as fungi. So soil habitats tend to be very microbe rich, and getting away from that soil habitat might have served a function of moving away from a very potentially pathogenic environment, because pathogens have a long history of relating to social insects and potentially being social selective pressures in their evolution of group living. And what I mean by that is that termites and other social insects have a lot of behaviors that they use in order to actually survive and be less susceptible to disease than they would be if they lived apart. Now, that's in huge contrast to us, as we are seeing now from, dealing with COVID virus. When we are closer together and interacting and, sharing fluids, as it were, that makes us sick. In contrast, termites are much better when they are interacting. So on the left-hand side is a photo of termites doing something called oral tropholexus, where they pass secretions mouth to mouth. They also do anal tropholexus, so mouth to anus passage. And that is because a lot of the fluids or feces that they pass to one another include antimicrobial factors. So if one termite has at least some immunity to a pathogen, they can potentially pass that to their nestmates. They also undergo allogrooming, where they pick off things like fungal candida off of their cuticles. And many social insects, including termites, also undergo something called coprophagy, where they either ingest dead individuals because when it passes through their gut, it becomes inactivated, or they bury it in feces, or they completely remove it from the nest and dump it outside. So lots of different examples of how it is more advantageous for these guys to be in groups. All right, so I kind of jumped the gun a little bit, but I also read in other animal species where they have studied or examined microbial concentrations in ground nesters versus arboreal nesters. It appeared that there were higher amounts of parasites in ground nests compared to arboreal nests. So I hypothesized that I would find differences in the microbial concentrations in an arboreal environment compared to their surrounding soil environment. So that was one of the major questions that I was trying to answer with my work. All right, back to the pretty locations. So I had the great fortune of being able to spend multiple years in St. John. In the beginning, this work was funded by something called the Earthwatch Student Challenge Awards Program. And then I was able to continue it a little longer after that program was no longer funded anymore, with faculty research grants and other smaller opportunities. But the species on this island is Nasutotermes acajutlae. So they are an arboreal nesting species, and their nests are actually some of the largest arboreal nests in the genus. So an example of one of their nests. Now from the outside, it looks more or less smooth, but inside the nest, what's going on are there are actually lots of convoluted tubules inside. Now these nests, if you were to kind of hack away at the outside a little bit, it's relatively easy to kind of peel off the outer shell. But something else of note is that these termites heal injuries in their nest very, very quickly. So any damage you do, they would build on top of it in less than 24 hours. So that posed another issue with trying to put anything inside their nest, because the soldiers also secrete a very sticky substance. It smells really good, but it is very, very sticky. So we actually had to lube our data loggers in plumber's grease, or else we would have never retrieved them from the nest, like ever. And we also tied a string to them. So we had to yank them out. And that was also a dangerous endeavor. But the island of St. John, some of the wonderful and advantageous things about working on this island is, for one thing, this is the one US Virgin Island that is primarily dominated by a national park. So it had large portions of it that were less disturbed and developed than the other Caribbean islands. And it's only about 12 square miles, so relatively small. So you could get across the entire island multiple times in a day with relatively little trouble, although driving on those roads is also quite an adventure. But within that very small space, there are actually 5 different habitats. So there was a lot of variety in environments in order to study how environment influenced these different factors that we were studying. So the environments that we looked at were woodland, mangrove. Mangroves are fun because you can see these really large prop roots from the mangrove trees, and everything underneath was soupy, squishy mud. So you had to be careful that, for one thing, you didn't lose your shoes. And also you had to kind of climb through and around all of those intertwined roots. The sparse habitat was essentially kind of beachline habitat. So any trees that were right along the beach edge was very sparse, and one of the tougher habitats for the termites to live in. And then at lower elevations, we would have dry forest. And in this picture, you can actually see one of our Tupperwares that we put our external data loggers in upside down, duct taped to a tree to try to keep the elements from getting to it. And then at higher elevations, now granted, elevations did not go beyond 1,000 feet. So these, even though these islands are somewhat mountainous, they're not too high. It was somewhat cooler. And by cool, I still mean that it's like 70 some odd degrees Fahrenheit, but very, very humid. So a little bit more like a tropical rainforest. And these, you've already seen some of them in the photos, but this is a selection of some of the SCAP groups that I have had the great fortune of working with over the years. So this is a program that no longer exists, which is very, very sad. But what the Student Challenge Awards program was a scholarship program for high school students that were gifted in the arts. And this program allowed them opportunities to increase their experience with science and math and things of that nature with field research by providing these field research opportunities. So the very first year that I went to St. John, I was involved with this program, and then following that first year, I actually became a co-PI of the project so that we could continue it for several years to come. So I actually did SCAP for 5 years. And our SCAP groups, they could either be mixed sex or just female or male. So we chose a group that was just females because, forgive me, but teenagers, you know, hormones, all that fun stuff. But also, who doesn't like to promote women in science? So rock on. So some examples of these wonderful women doing their field work, including collecting trail termites, coring into a nest, and then once we got the cores, we would spill out the contents to either collect termites and nest material, but also to find out if the nests were reproductively active, so whether they had reproductive termites in there. So Claire had been going to St. John since 1998. She actually used to work at the university that was on St. Thomas. So she already had many years of data even before she met me. But then once she met me, we also introduced environmental factor measurements. So things like nest ambient and soil temperature, nest ambient and soil percent moisture, light, nest volume, nest height, soil pH, all that fun stuff. This is an example of one of our data loggers that's about the size of a credit card, and you can actually see some of the termite nest carton that got stuck to it from being inside the nest. And this photo is an example, another lovely example of using the tools that you have to get at a nest, including our rental car. So, you know, sometimes rental cars are also needed, not just for driving. And this is our laboratory, AKA a cabin in the woods. So the unique thing about where we did our work, it was a lovely place called the Virgin Islands Environmental Resource Station, or VERS. And this station did have a somewhat elaborate marine lab, but in terms of doing terrestrial studies, the lab was not very developed. So there was some equipment that we left in the lab year round if it could withstand high temperatures, humidity, and salt. But that is very corrosive. So a lot of the material we just carried back and forth every year. So things like microscopes, a little mini centrifuge. I think one year we also brought gel pouring and construction capabilities. It was crazy. So lots of ingenuity in terms of how to do lab work when you don't actually have a lab. So I joke that lab work is also kind of field work, at least in this case. But we were able to do quite a bit and accomplish many, many things in very rudimentary situations. So these are some photos of me collecting trail and nest material. And I would bring all of that back to our lab and essentially wash the outsides of both the termites and the nest or trail material or the soil with a kind of sticky, mild detergent solution. And then I would plate a portion of each of those washes and try to culture the bacteria and fungi. Now, something to keep in mind is that this is very old school, right? Because not everything that's in the soil and the environment is culturable. So it's just giving us, you know, some of the picture of what's actually out there. Nowadays, molecular techniques are all the rage. So my old school techniques are not as cool anymore, but I still like them. I like to see things. I think it's more fun. So after a lot of plates and much, much work, we get a very busy graph, but I will summarize. So basically what I found was that overall, in all the habitats, pretty much, we were finding less bacteria and fungi inside the nest compared to outside the nest. So that was true of both the trails and the soil. The core was consistently lower. The trail and soil kind of went back and forth. So this told us something very important, which is that the internal nest environment has mechanisms in place to reduce the growth of these bacteria and fungi. So that is a very cool thing. So from other studies, we know that it might have a little bit to do with the different environment inside the nest, which I'll talk about in a moment, but also because of what the nest is constructed of, there are antimicrobial properties in the nest material itself. So reduced growth inside compared to outside. We were also able to identify some of these fungi, and I like to put them up because I think fungi are super pretty, but I'm also weird in that way. And this leads to another very funky graph. But I am hoping that even though this graph is incredibly busy, the point that I wanted to get across was that in the core of the nest, of the fungi we identified, there were fewer found within core samples than there were in trail or soil samples. So again, less fungi inside than outside. Another study that I did in collaboration with Claire Fuller examined how environmental conditions influenced the immunocompetence of our termites. So something that we're finding nowadays more and more is that with changes in climates, there's a lot of concern that this will change the ability of organisms to withstand infections. So that is one reason why this study was important, but the other reason why was because this was one of the few studies that actually measured this actively in the field. So a lot of immunity studies happen in the lab, but our lab is the field. So we got to do it outside right there. And I should mention our master student, Andrew, that also worked on this project. So this is a very blurry photo, but basically we had groups of termites that were exposed to fungus in these culture dishes, and we left them out in the field so that they would stay at either moist, cooler conditions at higher elevation or the dry, warmer conditions at lower elevations, and compared their survival. And we also swapped termites that came from those environments and put them in the opposite environment. Hopefully that makes sense. And when termites died, we would make sure that they died from the fungus that we gave them. So we would put them on plates to see what actually kills them. So this is Metarhizium anisopliae, which around here is actually a common soil fungus that insects are very susceptible to. Also in this study, we took very many environmental measurements, and we studied a couple of other immune parameters. So in addition to testing their susceptibility to fungus, we also looked at two main insect immune factors, one of which is overall lipid content. So they have immune factors in their fat bodies. So looking at overall lipid content can be indicative of immunocompetence, but also they have something called the phenyloxidase cascade, or PO activity, and that is tied to the production of melanin. So if you injure a termite, and I actually did this a lot because I would extract hemolymph or their blood from them frequently, they form a little melanized patch to kind of heal the injury. So that actually also happens inside their bodies when something invades. So they do this PO cascade in order to kind of help restrict and fight infections. So we did a lot of tests, but the moral of this story was that we were able to find a strong correlation between temperature, for sure, a little bit less humidity, but basically we did find a connection between the external environment and how susceptible they were to infection. And we also found that nest volume did play a little bit of a factor as well. And nest volume has been tied to the age of the colony. So that could factor into how susceptible or not susceptible they are to infection. So just some graphs. On the left, we have PO activity and how that relates to ambient temperature and nest volume. And on the right is total lipid content. And you can see some are prettier than others, but you can see some very nice trends in those lines. All right. So in that study, and also in the next study, we took a lot of data logger measurements, like a lot of them for multiple years. So I wanted to give you guys one example of the readout from a data logger. And the top one is the ambient outside conditions, and the bottom is the nest. And the point I want to make is how stable the inside the nest is. So termites maintain an internal nest environment that is both warmer and somewhat more humid than the external environment. And that is how they like it. So in 2014, Claire and I wrote a paper that literally said, termites like it hot and humid. And that is one thing I appreciate about Dr. Fuller, is we have a similar sense of humor. So to summarize, I just want to point out that after studying environmental data from multiple years and comparing the nests outside and inside, the external environment is overall warmer and more humid, and that trend continues across multiple years, which is pretty nifty. So before our study, lots of studies had been done on other termite nests, but none before had done very much with arboreal nests, and at least not for this many years. So something else we learned when taking all of this temperature and humidity data, we also tried to tie it to the presence of reproductives. So we did also find that in the warmer nests, we more likely found the presence of nymphs or alates. So it's a little bit of a toss-up of whether they are able to produce them better when the temperature is warmer, or a lot of that internal temperature actually comes from metabolic activity. So when they're producing reproductives, they actually produce a lot of heat. So it could also be tied to that. And then on the right-hand side, something that's really fascinating is that we also took temperatures of live nests versus dead nests that had been vacated versus the ambient temperature. And a dead nest with no termites can sometimes have a lower temperature than ambient temperature. So it just goes to show that that nest material actually does have a little bit of an insulatory function to it, and is able to kind of hold temperature in whatever that temperature happens to be. So what comes next? That is kind of a quick and dirty summary of many, many wonderful years spent in the Caribbean. But I am sad to report, which you guys probably, many of you might know, that several years ago, there were two devastating hurricanes, Irma and Maria, that completely destroyed Beers where I used to work. And this has me very, very heartbroken. So first off, the funding ran out. That was the first sob story. And secondly, the study site got destroyed. So the islands are still recovering. And as of yet, while other parts of the island have started to rebuild, as of yet, Beers is still completely overgrown and derelict. I have read that there were some applications for federal funding to rebuild, but I am not sure yet what will happen with my beloved island. So in the meantime, the other point I would like to make to end out my talk is that field research does not just mean going out to all of those fun, awesome places. Sometimes the fun, awesome place is right outside your door. So a little bit closer to home, I currently have several students doing field projects right on our campus at Virginia Wesleyan University, which we are very fortunate to have many acres of forested lands that are chock full of termites, which very much excites me because till a year and a half ago, I was still in the Northeast. And while we do have termites up there, they are definitely not as prolific. So it's kind of fun for me to live in the world of termites again. And I am probably one of the few people that kind of judges their places based on how many termites are out there. So the species that we have around here, the primary one is Reticulotermes flavipes, a strictly subterranean nesting species, and also others in that genus. And this is my student Molly, just the other day, collecting her first termite nest in the middle of February. So another thing I love about Virginia is termites all year round. It's good times. I have another student working on insect acoustic surveys in our old growth beech forest. So this on the left-hand side is a tree with a wild acoustics acoustic recorder. And we are trying to determine what type of diversity of insects we have in that old growth forest. And then field doesn't have to be just restricted to your research. It can also be incorporated into your classes, which I think I have some of my students here, so they know what I'm talking about. So I also try as much as possible to bring students out into the field. And one of our unique habitats around here are the cypress swamps, and we saw some beautiful rainbow swamps the other day, which were phenomenal. And with that, I just wanted to acknowledge both all of the people influential in my research and also funding sources, and I am happy to answer any questions.
Well, thank you very much. I will say that I very much enjoyed your presentation, some nice adventures and stories and relating the science to the adventures. I'm going to hold off on asking questions to give our audience a chance to do so, but remind them that you are free to unmute your microphone and ask questions or make some comments, and let's have a discussion.
All right. Oh, Claire is here. So I probably should give her credit. My collaborator, Claire, was in the audience. So that is very exciting. Yay, Claire. So John mentioned, I noticed you all did a fair share of climbing to obtain samples. What's the highest or most difficult sample you have ever collected? So unfortunately, I have no formal climbing training, which is actually one thing that I hope I can do someday in the future. So I think that one where I used the plastic chair might have been the tallest tree that I climbed. I think possibly. Although there may have been some slightly higher ones in St. John, as long as there were enough branches, I would be willing to climb it. And actually, that's another good thing that high school students are good for. They really like climbing trees. So even if Claire and I couldn't climb it, they could climb it for us. Another good question. How long did it take to collect all the data? So we would normally go, these SCAP programs tended to run, oh my gosh, it's been so long. I think they were 2 weeks with the students, but we would also add some time a little bit before and after. So we would try to spend at least a few weeks there. So not very long at all, but we did go there multiple years. So I think I was there 2004, 2005, and then 2007 through 9, and then a couple years after that as well, interspersed. So I think I went for at least 7 different years. Any other questions?
I got one for you, Marielle.
Sure.
So you mentioned you were on your way to Panama, hopefully. Did you ever get there?
I did, and actually, the picture with the plastic blue chair, that was actually from Panama.
Oh.
So I do actually have another fun story that is partly tied to my 1 year there. I got to spend 1 year, but then so much of my research was focused around the project in St. John, and I had funding from there, so I did primarily end up working there. But I had a fellow grad student, an undergrad, that went to Panama multiple years. They actually got some of those wonderful like 3-month research grants. So my friend spent 3 months in Panama, got all of this data, but it was post 9/11. So anyone that works in far off places, another lesson I will teach you is be very careful when transporting samples back to the US. Because she had 3 months worth of samples, some of them were live, and unfortunately TSA opened one of her canisters on the plane, tried to close it back up, but it did not follow proper USDA guidelines. So also USDA and TSA need to have a chat because they do not intermix very well. So unfortunately, was not resealed properly. So what happens, they arrive in New York City, folks go into the cargo hold to get the cargo, and there's termites walking around the plane. So I like to joke that's the next movie, not snakes on a plane, but termites on a plane. So yes, that is another downfall of bringing stuff back. I actually also collected a nest, and Claire got to enjoy that experience as well, of collecting a nest that had flying alates in it. We popped it into a tote, brought it on the plane, and then I hear my name called over the intercom, and they're like, please report to the blah, blah, blah. It's like, okay. So they're like, are you guys the ones transporting wood? I was like, yeah. They're like, it's infested with termites. I'm like, I know. That's the point. So yeah, that threw them off a little bit. And thank goodness the alates did not start flying out of the tote while they were sitting there holding it open. I was like, please close it, please close it, please close it. So that was a near miss, but thankfully they did let me keep my termite colony. Oh. All right, so another question, how would I expect the study site habitats such as mangroves and moist forests to be impacted by the hurricanes and flooding? That is an excellent question. So I have a dream to go back there, and unfortunately it has been several years now because it would have been super awesome to do studies immediately after the hurricane to see how that influenced the termites. But the amazing thing about these termites is they are very flexible and resistant. So I have a feeling the biggest difference would have been that the colonies that were previously in place, you know, a lot of trees fell down and probably a lot of them got destroyed, but that would not necessarily destroy the colony. They can rebuild those like within a year. probably less than a year. You know, we only went there in the summer, so we didn't get to see the in-between process, but we would be there 1 year and the nest would be in one place. And then we'd come back the next and the old one had died. And then there was a new one like just as big right next to it. We're like, what? So I have a feeling they're still going strong. The unfortunate thing is that, you know, these nests had been tagged for over a decade and we had been measuring them for over a decade. And so all of those original nests would have been displaced. So that long-term data set would have been interrupted, which would have been a huge bummer.
I have a question. I found it interesting that bacterial load and microorganisms essentially were lower at the core, in the core of the nest. Do you think this is due to them simply cleaning and pruning the nest, or do they do some sort of antimicrobial spraying?
I think part of it has to do with the nest material itself. So another individual that he used to be a postdoc in my grad lab, but now is at Towson University, Mark Bulmer, he has done a lot of work with substances called beta-1,3 glucanases. And this is a substance that the termites excrete or secrete that is built into their nest material, and it's antimicrobial. So they have antimicrobial factors built into their nest material in and of itself. And I think that keeps a lot of the growth down. But then also there are higher temperatures in the nest. So I think I forgot to point out on the data logger printout, but they keep their nests at 30 degrees Celsius or above. So their nests are super toasty, which for some bacteria can be okay, but for fungal growth is not as great. So a little bit of it has to do with the environment itself that they're maintaining their nest at. Claire is adding that we also did a study where we ground up workers and soldiers, and that, yes. So, after so many years in St. John, I don't remember everything we did, but we did do a study where we ground up workers and soldiers, and from that mush, we put it on fungal conidia, and it actually inhibited their growth. So those same antimicrobial substances are coming from the termites themselves. Any other questions? You could probably
tell us a little bit about how a conversation goes with someone that hates termites and how you can turn them around or attempt to turn them around to love them.
I am an excellent termite converter. I mean, perhaps it's my own enthusiasm about the termites themselves, but I have definitely converted some people over the years. So the girls that we would get in St. John, so the SCAT program didn't let you necessarily choose which trip you went on. So these were the poor suckers that like thought they were going to get to go on some fun adventure with like, you know, cute little pandas or I don't know, some other much more charismatic creature. And then they end up in the termites. Our only saving grace was that it was in the Caribbean. But by the end of the 2 weeks, I mean, you know, not all of them are going to be in love with termites, but we had them pretty convinced about how awesome they were. So I spend a lot of time promoting termites, and I think it works out pretty okay. All of my students learn way more about termites than they ever thought they would, even if that's not what the class is about.
I'll ask one more while others are thinking of questions. You know, you mentioned the aspect of the parameters inside the nest, and there was a question raised relative to the impacts of the hurricane.
Yes.
What about we hear a lot about global climate change and shifting temperature regimes. How is that likely to impact termites and their behavior and distribution?
So I think it is definitely going to influence the distribution of some. Now, the Reticulotermes genus that I work with locally, they are actually remarkably cold tolerant. I mean, I was finding them active on a 40-degree day and it was like super wet outside. And I was like, what are you doing out and about? You're crazy. These are also termites where most insects you can knock them out by putting them in the refrigerator, and it's kind of like their anesthetic. These guys would be walking around in the fridge totally fine. So, so termites are amazingly resilient, but I have a feeling with climate warming, we are going to see some species expanding their ranges. So in the Southeast, very unfortunately for Florida, they actually have the most instances recently of novel termite species that are invasive trying to make their way into the US from places like Mexico. So with climate change, that's probably going to become even more of a problem just because it's so easy for the termites to become established in such a nice, friendly environment. But conversely, there is a lot of concern for insects with respect to parasites and pathogens. So for non-social insects that don't have all of these like behaviors and ways of dealing with infection built in, they are predicted to actually have a lot more trouble because of more voracious infections as climates warm and change. So it's a little bit of a mixed bag, but provides lots of opportunities for me to do more research in the meantime.
All right, one last chance for questions. Going once.
Going twice.
All right, well, I don't hear any others. I want to thank you very much for taking the time to present with us today. I found it very interesting and entertaining. It was great, and I do want to take a moment to thank specifically Lainey and John for doing all of the work that makes these talks possible and working out the logistics. Without them, none of this would be possible. I also want to thank everyone for attending today's presentation, and I would like to put in an encouraging word to ask you to invite a friend. Come join us next week at the same time to